Welcome. Today we are going to be discussing evolution, specifically the evolution of HIV. Biological evolution is any change in the inherited traits of a population that occurs from one generation to the next. In other words, evolution is descent with modification. The theory of evolution started as an idea thought up by natural syngeologist Charles Darwin. Darwin came up with 4 tenets of evolution and natural selection by studying the species of finches he found on the Galapagos Islands. Darwin's 4 tenets are: more individuals are produced than can survive in each generation, variation exists among individuals, the variation is heritable, and certain traits are favored by the environment. These 4 tenets of evolution can be related to our topic today, the evolution of HIV. HIV stands for the human immunodeficiency virus. The human immunodeficiency virus is a very widespread virus. In 2007, it was estimated that a total of 33 million humans worldwide suffered from HIV, and of those 33 million, it was estimated that 3.1 million would die each year from the virus or related cases. The virus replicates in the host at extremely fast rates, and it has a long incubation period. HIV weakens the immune system by attacking T cells, which is a type of immune cell, leaving the host susceptible to other immune invasions, which can lead to acquired immunodeficiency syndrome, or AIDS. HIV is very dangerous because once the virus is acquired by the host, it remains with the host for life. There are 2 strains of the virus, HIV-1 and HIV-2. HIV-1 is prevalent globally and more dangerous. However, HIV-2 is characterized by a lower transmission and is not as likely to progress to AIDS, and is prevalent mostly in just West Africa. There are currently no cures for HIV or AIDS. The only forms of treatment involve suppressing the side effects. In order to determine a cure, scientists are looking into the virus's past for answers. It is thought that HIV originated by the evolution of simian immunodeficiency virus, SIV, which infects wild chimpanzees. Beatrice Hahn and Martine Peters charted the evolution of SIV. In 2003, they were able to demonstrate that the chimpanzee SIV was actually a mosaic virus consisting of viral components from 2 different SIV strains, the red-capped mangabe SIV and the red-spotted nose guinan SIV. Both viruses were able to jump from host to host by either sexual interactions or by contact with body fluids such as blood consumption. The particular order of cross-species jumps is not currently known, but what is known is that at some point a chimpanzee became infected with both the guinan and the mangabe virus, which portrayed a perfect scenario for the 2 viruses to mix and become a new, different virus. This new hybrid virus evolved to reproduce in a more efficient way than none of its viral parents could, infecting chimpanzees as far west as the Ivory Coast to East Africa. It was previously thought that SIV did not harm the chimpanzee host, but after further research it was determined that the chimpanzees did eventually get sick. At the same time, due to its capability to persist in chimpanzees for decades before causing them to die, SIV was eventually able to transfer to humans. It was then determined that there are 3 separate introductions of the SIV from chimpanzees to the human race. In the same way, it is thought that due to contact with chimpanzee blood fluids or their consumption by humans resulted in the different infection events that led to the development of the contemporary HIV. An issue that arises when trying to determine a cure for HIV is how rapidly it evolves. Scientists must determine a cure or vaccine that is able to replicate with HIV to keep up with its changes. The amazing thing is that there are people who are immune to HIV. Scientists have discovered that humans with mutations in an HIV gene, CCR5, which is a cell receptor that binds HIV to help it enter the human T cells. The mutation makes the receptor cells immune to HIV. Scientists are working to cause this mutation in people's CCR5 gene to keep HIV from being able to enter human T cells. In the beginning of 2015, the FDA approved the start of a new phase 1 study, which could consist of a multi-year 3-stage process of human trials to test a method of disabling the CCR5 protein. If this method works, this will be able to be the world's first functional cure for HIV.
created using Powtoon.